So now it's my pleasure to introduce Romain Lopez, who's at Berkeley, and he will switch to a different topic. So he will talk about modeling gene expression.
Hi, everyone. So I don't know very well the pain of organizing BayLearn, so thank you for that. But the pain I know is the pain of commuting from Berkeley. But today I will talk about applications of deep learning in single-cell RNA sequencing, so mostly in biology. So I'm very fortunate to be working with these people. Jeffrey, who is my variational inference specialist. Michael, who is my biology specialist, though he's in physics, but yeah, anyway, that's a side talk. With Mike, who is a basketball specialist. And sorry, that was easy. And with Nir, who is my computational biology advisor. I mean, yeah, these two are my PhD advisor. So yeah, what I want to talk about is a new technology that enables to look at a single cell, the expression of mRNA. So you know, you have the DNA gets expressed into mRNA, and now we have a new experiment that that got pretty popular from 2015 that enables you to look at the single cell level, the expression of every gene. So ideas like before, we were looking at bulk data. What bulk mRNA sequencing was, ideas like you want to look at the constituents, chemical constituents of fruits, let's say this. And for this, you would have to just like mix them together, and then you look at the chemical constituents. You can do some tests, you can do whatever you want. But this was the level of experiment that we could do before. Now with single cell, we can recover back all the fruits. So what are these fruits? Fruits are cell types. And we can get at every fruit what are the chemical constituents for them. And this is like pretty encouraging, and people are, biologists are making like really new discoveries with it. And so it's a pretty exciting area. I will try to explain also what is an exciting area for machine learning and what we can do about it to enable data analysis of this as large scale. So why single-cell RNA sequencing experiments yield large data sets? Because we can look at every single cell, and now a big company called 10x has enabled the workflow for the experiment to scale to millions of cells. Because we look at the expression of genes, so in my fruits, the chemical constituents of the genes, then I can understand exactly like how many times these genes have been expressed in the cells. And in the human genome, there are like tens of thousands of genes, and so this data is high dimensional. Then we can understand what is the noise of the data. Okay, I come back to this matrix. All the entries in the matrix are counts, so that to be clear. Now, what if we want to build a generative model for these counts? Where does the counts come from? What are the properties of the distributions? So genes can be either expressed or non-expressed, depending on like Markov jump process, because these are exponential waiting time, and the gene counts can, if it's on, the mRNA can be produced or can be destroyed, and when it is off, it can only be destroyed. So this kind of kinetics, when you look at over time the distribution, and you just forget about the time, what you recover, because we don't get to see the time in the experiment, right? We just have like a bunch of cells, and we just don't know in which state they are. And we observe the distribution, and it's a Gamma-Poisson, or a negative binomial. But let's say a compound between a gamma and a Poisson. And then the other thing about the noise, to generate these counts, is that because in each cell we can capture only a finite portion of the mRNA transcripts, we cannot get so here we cannot get all the colors, which means we cannot get all the genes. So we're going to have more zero in the data, if you want. So overall, we can model this as almost a uniform fraction of the data, plus some genes that are not captured, so more zero. Then what do we do with this? Yeah, we can do something. But before, what people did with that? So what people did with that is they try to do like, okay, so approximate data as like continuous data, continuous distributions, and they make algorithm with Bayesian statistics, but they need to do sampling, so it's really not scalable. And they make linear assumptions because there is nothing else like you might want to think about if you do some simple model. And people are afraid of neural networks because they think it's a black box, I cannot interpret anything. But the idea is like if you incorporate the neural networks into a Bayesian framework, then you can do something interesting. And so this is the algorithm I propose, which is called single-cell variational inference, which is a deep generative model for this kind of data. So this is generative distributions. So essentially, when you look at the graphical model, on the left, you have the zeros, because you draw from a Bernoulli and then you apply a zero mask, so you zero the data. And on the right, it's a compound between the gamma and the Poisson. So it seems relatively easy, and the prior is just a normal zero. Actually identifies the zero that come from the technical noise and the zeros that come from the biology better. And we also, when there is a zero, we can compute what would have been the value without the zero. We can compute it also better compared to other algorithm. Even like MAGIC, that is like the standard algorithm that is used for now in computational biology. And then what we want to, biologists love clusters because each cluster is a cell type. So the idea is like our latent space also like capture better the clustering. We use a silhouette score for that. This is how biologists would try to think about the data set. They would take a latent space and then use a TSNE like to try to lay out and visualize the data. So this is also pretty encouraging. But now the real idea is like we have a Bayesian generative model, and because we have this, we can ask questions with the data. And this is the most important point. So the first question we tried asking was, if I take two clusters, like cluster of B cells and cluster of dendritic cells, then essentially what are the genes that are differentially expressed from one to another? And so here we have like a data set, looks like this, population A, population B, and what are the genes that are differentially expressed from one to another? Okay. And the idea is like, because we have a Bayesian framework, we can do this. We just write a hypothesis. So the W would be like the mean of the expression of the genes conditioned on the non-zero. So we can remove all the technical noise in the data. And we just write a bias factor, like the posterior of the hypothesis based on having seen the data. And what we see is like, so we compare this with another test that is used all the time in single-cell RNA sequencing. And we compare these two tests with a biological ground truth given by a completely different experiment. And we see that we yield like genes that are more correlated with the other previous experiment that is like the biological ground truth, which means essentially we select better genes. And so this is like what we're really excited about for now. What we want to think about next, because we have something that is really large scale and that nobody else can do, we are thinking about experiments when we can integrate data across different, it can be different species or different patient phenotypes. And right now, I'm working with, I have a collaboration with a neurologist in Germany that has data sets from cerebrospinal fluid cells, from multiple sclerosis patient and like a control group. And we have like 10,000 of cells for each patient. We have 12 patients, and we're trying to integrate that and redefine what is the notion of like having multiple sclerosis, and so it's something I'm pretty excited about. And yeah, so that's all. You can look at our manuscript on, not a manuscript, but extended abstract on archive. And thank you very much.
Thank you, Romain. Do we have questions for Romain? And if the next speaker can set up while we actually work on the question. Yeah, I was wondering, so how do you, so basically you get like, so you compare, unless there's a question from the audience. So I was wondering how you compare, so you had like these two settings where you had like, one way to measure and your proposed approach. And then you get different errors. So in your case, I guess you have like estimation errors. Yeah, so yeah. And maybe some other method would have like, I don't know, measurement errors due to. Yeah, yeah. So how do you build trust when you work with biologists to say, well, my method is like reliable. So what is your strategy?
Okay. So the idea. Oh yeah, that's good. The idea is like essentially with single-cell data, there is a lot of noise, so you cannot trust anything on single cell mostly. But because we use another experiment that is more expensive with less data, so less noise, and that this data set is like 10 years ago. So people have been analyzing a lot, and people usually take these genes to write biological paper. So we can kind of trust this, but I wouldn't trust like anything on this data because we don't understand it very well yet.
I see.
Yeah.
Okay.
Okay.
Thank you.
Thank you very much.
Thanks.